So this is photosynthesis and everything you need to know about photosynthesis for AP biology. So in previous chapter we talk about because of the law of thermodynamics, right? Living things as is being ordered and organized, it really requires an input of energy in order to stay ordered and organized. So where does that energy come from, right? So you should be aware of the process of photosynthesis and how the process can store light energy into food. So we're going to go deeper into this. So first, a few key terminology. We can basically divide living organisms into two types based on how they obtain energy. The first type is called autotroph, in which they can generate energy from inorganic sources. From inorganic sources. So they can get it from non-living things. And they can do it in two ways. The one that we are more familiar with is photosynthesis, in which they store light energy into chemical energy, which we commonly know it as food, right? So these foods are, in general, a type of carbohydrates, such as glucose. So you can see right here, we have all these organisms that can do photosynthesis. Common knowledge is only plants do photosynthesis, but that's not true, right? We are more familiar with plants being able to do photosynthesis, but there are also other organisms that can do photosynthesis, but they are not plants. For example, algae, they are multicellular. They are a type of protist. They are technically not plants. Then we have eukaryotes that are unicellular, such as Euglena, which is also a type of protist, right? So they do have the ability to use light energy and convert that light energy into chemical energy. But there's also um, um, another thing is not only can eukaryotic cells do photosynthesis, right? Protists and plants are eukaryotes. Prokaryotic cells can also do photosynthesis. Cyanobacteria and purple sulfur bacteria, they do not have chloroplast, but they do have the ability to also use light energy and store it into chemical energy. So the idea here is that photosynthesis is not unique to plants. It can happen in prokaryotic cells that do not have nucleus or chloroplast, or in eukaryotic cells, it can happen in unicellular organisms or multicellular organisms, such as plants. Another type of autotroph is can store energy through the process of chemosynthesis. So in this case, they convert chemical energy that are of inorganic sources, non-living sources, into chemical energy that is usable for them, right? So these kind of organisms usually are found deep underwaters that they do not have access to light, but they are very close to volcanoes, and the volcanic ashes contain chemical energy, and they do have the enzymes to break down those inorganic sources and convert them into energy that they can use. So deep underwater, there's no sunlight, there's no plants, there's no organisms that can do photosynthesis, but there is a food web down there that is driving as well because the producer or autotrophs convert chemical energy into another form of chemical energy that they can use. So next we have heterotrophs. So heterotrophs cannot produce their own energy from inorganic sources. They have to obtain their energy by means of injections of other organic sources, essentially other living things. So we can subdivide heterotrophs into two types, consumer and decomposers. Consumers, as you're aware of, are like animals, right? They eat food and then they can break down the food and obtain the energy from it. We also have decomposers in which they can break down organisms that are already dead. They break down dead organisms and then release the nutrients into the ecosystems. And through that process, they obtain the energy from that. Heterotrophs, as of right now, is not the main focus of this chapter, so we'll just skip it and we will revisit this concept later. So the main focus is actually photosynthesis, right? The most popular organisms that we are familiar with is plants, and the organs that they use to do photosynthesis is leaves, right? So the process of photosynthesis is they're using carbon dioxide from the air, CO2, and water, H2O, from the roots, and then they can convert it with the use of light energy into sugar or glucose, C6, H12O6, and oxygen gas, O2. CO2 is obtained from the atmosphere, so in the air. H2O is obtained from roots. If you recall back in Unit 1, we learned that water, because of hydrogen bonds, is connected and can be pulled outwards all the way to the leaves where photosynthesis occurs. So photosynthesis occurs in the leaves. And they produce the sugar, which they store inside the plant's body for further use to obtain energy and release oxygen gas, and the oxygen gas is released into the atmosphere in which other organisms can utilize this. So if we summarize the whole process of photosynthesis, we have a chemical reactions here, and the chemical equation is CO2 plus H2O with the input of light energy makes glucose, C6H12O6, and oxygen gas. You need to be familiar with this equation, and you do not need to know how to balance the chemical equation. You do need to know the relationship between these chemical compounds inside the chemical equation. So CO2 becomes C6H12O6 through the process of light-dependent reactions. And during light-dependent reaction, CO2 is being reduced to become C6H12O6. Reduce is a term to describe a chemical that undergoes reduction. During a reduction reaction, a compound is losing electron. Sorry, gaining electron, my bad. Gaining electron. And subsequently, it also gains energy. How do you keep track of that electrons? The trick here is to look at their hydrogen. If they gain a hydrogen, see that CO2 doesn't have a hydrogen, and then C6H12O6 has a hydrogen, it is itself gaining electron. So if you notice a chemical compound gain hydrogen, it essentially means it gains electron. So H2O here is being converted into O2 and is losing electron through the process of light. Sorry, I just noticed I wrote this wrong. So CO2 gets converted into glucose through light-independent reaction, and then H2O gets converted into oxygen through light-dependent reaction. because I've been so used to talk about light-dependent reaction first and then light-independent reaction, I make a mistake here. Okay, so water gets converted into oxygen through light-dependent reaction, and water's molecule is oxidized to become oxygen gas. So oxidize is describing the process in which H2Os undergoes the process of oxidation, and it is itself oxidized into oxygen. Its water itself is oxidized into oxygen gas. So oxidation, during the process of oxidation, the molecule is going to lose electron, and subsequently it lose hydrogen, and itself will lose energy. So together, reduction and oxidation always goes hands in hand, right? So if inside a chemical reaction, if a compound undergoes reduction, the other compounds will have to undergo oxidation. So the key term here that you need to remember here is which molecules is being reduced and through what process. CO2 is reduced to glucose through light-independent reaction, and water is being oxidized through the process of light-dependent reaction. Together, light-dependent reaction and light-independent reaction forms the basis of photosynthesis. Next, we need to keep track of how the atoms are moving in the chemical reaction. So remember just now we talked about water is converted into oxygen gas through light-dependent reaction. And then CO2 is being converted to glucose through light-independent reaction. Notice that the glucose, carbon, and oxygen purely comes from CO2. Oxygen gas, the oxygen atoms purely comes from the oxygen of water. So what it means is even in a chemical reaction, we write CO2 becomes H2O and then C6H12O6 and CO2. The oxygen from the carbon dioxide never goes to oxygen gas. The carbon and oxygen from the carbon dioxide just strictly goes to the glucose. The oxygens from the water strictly becomes oxygen gas. So how does the plant's leaf able to do this? So if you zoom into the plant's leaf, you can see plant's leaves are made of many cells. The primary cells are the cells over here, and you can see that there are lots of dots in there. That dots is representing chloroplast. So in the cells of leaves, they are very rich in this organelle called chloroplast. In Unit 2, you learn that chloroplast is the main organelles that undergoes photosynthesis, right? So this is where photosynthesis occurs. You also learn that chloroplast has three membranes. The outer membrane, so the outer chloroplast membrane. So I'm gonna put it in the number here. So number 1 here, that's the outer membrane of the organelles. So that's the outer chloroplast membrane. Number 2, this is the inner chloroplast membrane. The in-between space between the outer chloroplast membrane and inner chloroplast membrane, that middle region is the intermembrane space. So these three things in the chloroplast are not the major focus of the functions of the chloroplast. What you need to pay attention to is the structures and locations inside the inner membrane of chloroplast. So the inner membrane, the space within the inner chloroplast membrane, that's kind of like the cytoplasms of the chloroplast organelle. That's called stroma. Then within this stroma, there floats many things are floating in science, right? We learn about the DNA that is circular in the chloroplast because of endosymbiont theory. There's also its own unique ribosomes that are floating inside the chloroplast that can utilize the DNA inside the chloroplast to make protein. Then you have this disc-like structure, which I like to describe it as the choco pie inside the choco pie. If you don't know what choco pie is, it's just Asian snacks that kind of thylakoid kind of just look like it. So it's a disc-like structure which is also made of membrane. So this disc-like structure, they are called thylakoid. Since they are also made of membrane, right? That thylakoid's membrane is literally called thylakoid membrane. Then the space inside the thylakoid membrane, because it's made of membrane, so you can imagine that there's kind of like the cytoplasm within that membrane, right? So that region, that fluid region, that's called lumen. So thylakoid is made of thylakoid membrane and that fluid inside the thylakoid membrane, lumen. You do need to remember the names of these locations, such as stroma, thylakoid, and lumen. A stack of a thylakoid is called a granule. So all the stack structure right here, that's called a granule. So in terms of the location, right, how do we fit light-dependent reaction and light-independent reaction into each locations of the chloroplast, right? So let's review. Photosynthesis has two reactions. It is the light-dependent reaction and it converts H2O into oxygen gas. This process requires the input of light, so without light energy, this reaction will not occur. So therefore, this reaction is called light-dependent reaction. It depends on light in order for the reactions to occur. So without light, waters will not get converted to oxygen gas. Oxygen gas will not be produced and released out of the plants. This process happens within the thylakoid structure, specifically the membrane of the thylakoid. Then we have light-independent reaction. This reaction is also known as Calvin cycle. Hence, you see these reactions happening in a circular manner. It is a biochemical pathway. It means it consists of multiple steps of chemical reaction. Light-independent reactions takes in CO2 and converts it into glucose. And this process happens in the stroma, which is outside the thylakoid but within the inner membrane of chloroplast. Okay? So here you can see we have our organelle chloroplast. To review, H2O becomes oxygen. So this process is a oxidation reaction. And CO2 get converts into glucose. That is a reduction reaction. Take home point from this slide. Remember which location does which reaction occur. So we're going to take a little bit of detour, talk about light as an electromagnetic spectrum. So light has two particular properties, which is wavelength and energy level. Light exists in multiple spectrum, like it goes from very short wavelength to very long wavelength. And there are many different types of light. Only one specific regions of the wavelength range is the type of light that we can see, and that's called visible light. So the visible light range from 380 nanometer to 750 nanometer. And it is exactly the color of the rainbow, from purple violet all the way to orange red. Purple violet has very short wavelength, but very high energy. Red and orange red have very long wavelength, but they have lower energy. So how does this relate to photosynthesis, right? Because photosynthesis uses light as a source of energy. The chloroplast the chloroplast contain a particular type of protein called pigment. So pigment is a type of protein that can absorb light at a specific wavelength, essentially at a specific color. Sometimes some pigments are better at absorbing a certain type of color, and the color that they can absorb will then convert that energy and excite electron. So absorb light at a particular wavelength is used to excite electrons. Excite electron means the electrons goes from ground state to excited state. Ground state to excited state. Ground state has a lower energy. If it gets excited, it will have a higher energy. Essentially, what we're saying here is electron gain the energy from light. That's why they get excited. Spectrum of lights, if it doesn't get absorbed by the pigment, it will then get reflected. So reflected lights are essentially lights that do not get absorbed. And that light color is what we see. So here I'm drawing an eye right here. We're seeing chloroplasts and plants in general green because of the reflected green light. This particular pigment is called chlorophyll. So example of a pigment is chlorophyll. Chlorophyll is really good at absorbing blue and red light, right? So absorbing blue, violet, and then red, orange light. So what's left is they're very bad at absorbing green. No green light is absorbed. So therefore, that green light is reflected. And that's why we see plants as green. Chlorophyll is not the only pigment in plants' chloroplasts. There are multiple types of pigments that can be found in the plant cells. There are also organisms that have different types of pigments that contributes to the type of color that we see them, right? So make sure you remember any light that we see are actually lights that they don't use to excite the electron. So if you see purple color, it means that they don't absorb light. They don't absorb purple color. It means purple color is not useful for them to excite the electron to make the electrons having a higher energy. So here, this is a graph that you need to learn how to read. The x-axis is the wavelength of light. And the y-axis is the absorption of light by chloroplast pigment. So notice that there are three types of pigments that are shown in the diagram. We have chlorophyll A, chlorophyll B, carotenoids. So notice that chlorophyll A, there are absorption level, as seen the y-axis, that are really high, around 400 nanometers to 450 nanometers. Then it has some sort of absorption somewhere around 650 nanometers to 700 nanometers. Then we can do the same thing for chlorophyll B. Chlorophyll B, it has very high absorption somewhere around 470 to about 580 nanometers. It also has a little bit of absorption somewhere around here, which is around 630 nanometers to about 650 nanometers. And then we have carotenoid as a pigment. It has very high absorption, somewhere from 400 all the way to 510 nanometers, but then it cannot absorb any light that is past 510 nanometers. Notice that the color of the graph kind of represents what the colors that you see on with your naked eye. Carotenoid, because it cannot absorb any color past 520, it appears orange because it cannot absorb colors that are like around orange color. Chlorophyll A and chlorophyll B appears green because they're pretty bad at absorbing energy around this region. So if you combine all these three pigments activity, you get an action spectrum. Essentially, it is the total absorptions of all different pigments, and we can kind of see how it correlates to photosynthesis, right? So if you have lots of absorptions, you will be able to absorb the light, and light-dependent reaction will occur, and it will produce oxygen as a measurement of photosynthesis, because photosynthesis produces oxygen. With lots of absorption, based on what we see here, right? There's lots of absorptions here. It means photosynthesis can occur. Therefore, you're going to observe lots of oxygen produced at that region due to high absorptions. Regions where there is no absorption means they are unable to produce as much oxygen gas, so the rate of photosynthesis decrease. So now let's move in to photosynthesis first reaction, which is our light-dependent reaction. Light-dependent reaction happens at the thylakoid membrane. So first we have to orient ourselves, like what exactly are we looking at? So we're looking at the thylakoid membrane. Membranes are made of phospholipid bilayers, but within the bilayers, we have many different proteins. There are a few proteins here that we need to pay attention to, right, which we'll talk about later. First, orient ourselves first, because this is a thylakoid membrane. It divides the region into two spaces. This region over here is what we're looking at right now. That is a stroma region. And then this region over here, that is inside the thylakoid. So that is our lumen. Stroma has a lower concentration of hydrogen ions. Lumen has a much higher concentration ions because, relatively speaking, lumen has a smaller space. The first protein that we're going to focus on is photosystem. So this protein here, this is photosystem 2. Photosystem 2. So a photosystem is a protein complex And within that protein complex, it contains the pigment chlorophyll. It doesn't have to contain chlorophyll. It can contain other pigments as well, right? The major one is chlorophyll. So it is a protein complex that contains pigment. And just now we said that pigments can absorb light energy. So photosystem can absorb light energy. When light energy is present, so here I'm going to draw a lightning to represent the presence of light energy. Photosystems pigment is going to excite the electron. So there exists an electron here, so which I'm going to write a symbol E. The E here represents electron. The H plus represents the proton. Electrons will get excited because of pigments. So I'm going to draw an arrow pointing out saying that the electrons has been excited. And I'm going to draw this bright line around here to represent an excited electron. This excited electron is due to pigments in photosystem 2 absorbing the energy from light. Excited electrons will get shuttled by the surface protein across the membrane. So it's just going to start moving along the membrane and it gets shuttled by the protein. The name of the proteins that shuttles the electron is not that important, so we will skip it. I will not to say it's important, but it's like not something that you need to pay attention to in AP Bio. Okay. During this process, the energy from the excited electron is being transferred to the second type of protein in the membrane, and that protein is called proton pump. So, because the energy is lost to proton pump, the electron is reverted back to its ground state. So you see that sparkling line around the electron is no longer there. The electron loses its energy. The energy is transferred to proton pump in order to do active transport of proton, right? Remember, when you see pump, it indicates that molecules being moved from lower concentration to higher concentration against its gradient. So we have an active transport of proton in order to build the proton gradient, which just indicates that there is a differences in the concentration of proton. In this case, lumen has a high proton concentration, and stroma has a low proton concentration. And you do need to know that and remember that. Then, as you have more and more protons, so before we talk about protons, we're going to continue follow the route of electron. Then the electrons will get shuttled to photosystem 1. So electron enters into photosystem 1. Photosystem 1 has pigment, so it will excite the electron to get into excited states again because of the presence of light. So a few differences here that you need to notice is first, the electron received from photosystem 1 has already undergone this process of shuttling the electron. This process is called electron transport chain. The reason why this is called photosystem 1, because this is the first photosystem discovered by scientists, and then later they discover a second photosystem. But in terms of the route of how the electron moves, electrons moves from photosystem 2 then to photosystem 1. Then this electron that's getting excited will now get shuttled to the next protein. Eventually, it will reach a protein that will convert this electron into an electron carrier. So not convert, it will transfer this excited electron with high energy to an electron carrier. And that electron carrier is NADP plus. So NADP plus will be converted into NADPH. So NADP plus, you don't have to know the full name of it, but the full name of it is nicotinamide adenine dinucleotide phosphate. It has two forms, the reduced form, which is NADPH, and it contains that electron that has high energy. Right? Remember when we say there's an H, you need to envision that there is electron there. So this electron is the one high in energy. So it comes from the NADP plus. There's an oxidized form. It does not have that energized electron. This process happens because of an enzyme called NADPH reductase. So there's an enzyme here that transfer the electron to NADPH. So now we're going to focus on the proton gradient. As you will learn in Unit 2, right, the high concentration of protons is going to build up pressure and potential in it. So the hydrogen ions has to move back and diffuse to the low concentration of protons region. But protons cannot pass through the membrane because proton is an ion, it's positively charged. So it needs a channel protein in order to do so. Hence, the protein that helps to do it is ATP synthase. ATP synthase is a channel protein that allows the diffusion of protons from high concentration back into low concentration. This flow of protons will turn the ATP synthase and convert ADP and inorganic phosphate into ATP. ATP synthase is an enzyme, so it will speed up that chemical reaction of combining ADP and P into ATP. And why does it have the energy to do so? Because it is converting the potential energy of this proton gradient into a chemical energy stored in ATP. Okay. So now the electrons have left the membrane, the ATP is being produced and is storing the energy. As you can see, right, ATP is storing the energy because of the proton gradient. The proton gradient is able to be built out because of the loss of energy from this excited electron here, because the electron is getting excited by photosystem 2. But where does the photosystem 2 get the electron once the electron has left the membrane? That's why you need to replenish this electron here that has left the membrane into NADPH. So here comes water. Right? So here comes water. So water will come in and get oxidized to become oxygen gas. This process lets water lose its electron. So the electron from water, right, because water has H, right, it becomes O2, no longer have H. That electron enters photosystem 2 to replenish the lost electron. So in summary, right, we can say that the role of water in this light-dependent reaction is to donate electron. They are the electron donor for light-dependent reaction. Okay? So, to summarize, right? Let's review back again. We have two reactions that happens in photosynthesis. The first one is light-dependent reaction. It happens in the thylakoid membrane. Thylakoid membrane. It converts water into oxygen gas to replenish the electron that is being excited by light because of the presence of pigments. Pigments absorb the energy from light, excite the electron. As water donates the electrons, that electron is being absorbed and taken away by NADP plus and then converted into NADPH. So now pay attention to the electron of water. The electron is flowing, right, through light-dependent reaction, and it is now in NADPH, as is in a form that has high energy. Because that electron did not go through electron transport chain to do active transport of the proton. The proton gradient that's being built out then can convert ADP into ATP and store the energy that was originally built from the proton gradient and store it in this form, ATP, right? So energy ATP has a much higher energy than ADP. So now the ATP and NADPH are being used by light-independent reaction. ATP has that high energy, NADPH has the electron that's high energy, and the role of light-independent reaction is to store all this energy from the two molecules using CO2 and store it into a compound, which is glucose. So by the time this light-independent reaction has that, by the time glucose is made in the light-independent reaction, it would have obtained the electron that is high in energy. It will also obtain the energy from ATP. That's why glucose is called the storage. Sorry, not storage, the source of energy, right? As ATP is reverted back into ADP, and NADPH is being reverted back to NADP plus. So here you can see the role of light reaction and the role of light independent reaction. The role of light reaction is really to store the energy from light into ATP and NADPH. You can also see the role of light independent reaction or Calvin cycle. It is to store the energy from ATP and NADPH into glucose using CO2. So now let's dig deeper into the light independent reaction. So in light independent reaction, because it is also known as Calvin cycle, it is a biochemical pathway that exists in a cycle. A cycle essentially means that the reactant that you use at the very beginning of the chemical reaction will eventually be regenerated as a product, and that product can be used again as the reactant. So this particular product and the reactant is called RuBp. RuBp stands for 1 ribulose 1,5 biphosphate. Okay. It's short for RuBp, and we all know what it is. It is the reactant and product of light-independent reaction. You need to know what the name of the enzyme is, and that enzyme is called RuBisCO. So RuBisCO is an enzyme, and it initiates the first step of Calvin cycle or light-independent reaction. The first step is combining RuBp with 3 carbon dioxide molecules and convert them into 3-phosphoglycerate. This name of the chemical here is not that essential to your understanding of the concept of AP biology. You can just think of it as some sort of organic molecule that stores CO2. Okay? So the CO2 from the environment combines with RuBp because of an enzyme called RuBisCO. This process is called carbon fixation. So during the process of carbon fixation, and we call this as a carbon fixation, not to say that the carbon dioxide does anything wrong. It just means that CO2 at that state contains a carbon that we cannot use directly, so it needs to be fixed. Fixing just means it converts into an organic form. An inorganic converts something that's in an inorganic form into an organic form that we as a living organism can use. Then we move on to the second step of light independent reaction, which is reduction. During reduction, inputs of ATP and NADPH is going to form G3P, which is the organic molecule used to make glucose and other organic molecules for life. G3P. A reduction requires the use of ATP and NADPH because ATP provides energy. So that energy is input into G3P. And NADPH is providing that excited electron, right? Remember that energy, that electron high in energy. That electron is in the H right there. So that's why as NADPH get converted into NADP plus, that electron enters G3P. So now, as you see G3P, it has that excited electron. It also has the energy from ATP. So now G3P can be used to make glucose. So here the point is that glucose is actually not directly made by light-independent reaction, but light-independent reaction makes the major ingredient called G3P that will be used to make glucose and other organic compounds, such as lipid and protein. So now you have that G3P. Then you need to remake that RuBp again so that this process can occur again. So the third step of light-independent reaction is RuBp regeneration. This process does need input of ATP in order to provide the energy needed to regenerate RuBp. So to summarize, light-independent reactions, there are three steps that you need to remember in sequential order. Carbon fixation using this enzyme, RuBisCO, very important. Make sure you remember the name of that enzyme. Then reduction, in which ATP provides energy and NADPH provides the electron. These two molecules comes from light-dependent reaction. Then as in light-independent reaction, if you make the ADP and NADP plus, they will get recycled, they will get reused by light-dependent reaction. The third step of light-independent reaction is RuBp regeneration. You remake the RuBp. This process keeps repeating. So let's put everything together, right? In plant cells, there's the organelle called chloroplast. There are two chemical reactions, the light-dependent reaction, and the light-independent reaction. The light-dependent reaction, according to this arrow, here I have to pay attention to what the molecules are. So this is CO2. So light-independent reaction takes in CO2. Light-dependent reaction takes in H2O. And they make oxygen gas and glucose. But the glucose is just storing the energy. It doesn't get converted into the energy for all the cellular activities in the plant cells. Plant cells, as a eukaryotic organisms, also has mitochondria. So mitochondria as an organelle will use glucose to provide the energy to make lots of ATP, and it will, as a result, remake water and CO2 so that the water and CO2 can be reused by chloroplast through the process of photosynthesis. Mitochondria, the process of using glucose and oxygen to get all the energy in ATP form and then produce water and CO2, the role of the mitochondria is undergo cellular respiration. So that's how you can see the connections between chloroplast and mitochondria. These two organelles work side by side in plant cells. Plant cells use chloroplast through the process of photosynthesis. Stores the energy from light and then put them in glucose. Then mitochondria has to access the energy through the process of cellular respiration, take in the glucose, break it down, release water and CO2, but also makes lots of ATP. And these are the energy in the form of ATP is the form of energy that can be used by the cells to do all kinds of cellular activity. For example, active transport. And that's the relationship between these two organelles. And we will go deeper into cellular respiration in the next chapter. That's it. I hope this video is helpful.